for the chromosome registry. And that was around when the vaccines were first available to the general public. So this is an update on where we are with COVID-19 vaccines as of December of this year. I've been in practice in allergy and immunology for 14 and a half years. I practiced in Ocean Springs, Mississippi, and in Biloxi. And I've been a member of the Chromosome 18 registry for about 12 years since Lewis was born, and we found out that he has chromosome 18p minus. So Lewis would like to say hi to everyone. Say it.
Hi.
And we'll go ahead and get started with the presentation. So I don't have any disclosures. I'm not affiliated with any of the pharmaceutical companies that have developed or the vaccines. And I'm mainly going to focus on the vaccines that are available in the United States. This information is constantly changing, and I just don't have time to follow up with all the vaccines that are available in other countries. So as of yesterday, here we are with our numbers globally. It's over 266 million cases of COVID-19, which certainly is higher than that, more than 5 million deaths. And globally, there's actually been more than 8 billion vaccine doses that have been given. In the United States, looking at 49 million cases, almost 800,000 deaths, and around 470,000 million vaccine doses that have been given. So how do vaccines work? Well, we're introducing a foreign material that causes an immune response, and typically it's antibodies that are most important. And these antibodies search for that foreign substance and prevent infection into the cells of our host cells. Let's see. Okay, so this is a cartoon that kind of just is a drawing of an IgG antibody. IgG antibodies have two binding sites for antigens. In these binding sites, it's sort of like a pocket where an antigen fits tightly. And so each antibody that's produced is very specific for a certain type of antigen. In this case with COVID-19, it's the spike protein, and there are several areas on the spike protein that antibodies bind to. There's more than 30 actually. So with SARS-CoV-2, most of the vaccine development has been focused on the spike protein. The spike protein is actually what binds to a receptor on our allergy cells, which is called the ACE2 receptor. That allows the virus entry into our cells and where it replicates. On the left side here, we have classic forms of vaccines that have been around sometimes, in some cases, for many years. And on the right side, these are the newer platforms that we have. And this is where we're focused on now with what's approved in the US. So at the top here, we have the viral vector vaccines. In the US, we have the Johnson & Johnson vaccine, and other countries, the Oxford AstraZeneca vaccine is approved. This is an adenovirus, which normally causes the common cold. This particular adenovirus cannot replicate, and so it cannot cause disease. And it's manipulated to express the spike protein that's found on SARS-CoV-2. And down here we have an RNA virus. There's two of those approved in the US currently. It's the Pfizer BioNTech and the Moderna vaccines. This encodes for RNA, which translates to a protein, which is again the spike protein that causes an immune response. So this is a cartoon showing inject mRNA vaccine. This is in a liposome that is absorbed into our cells. Once RNA is in our cells, it goes directly to a ribosome where it is translated to the spike protein, which then is expressed outwardly on antigen presenting cells. It's actually presented to T cells first. T cells present it to B cells. B cells then make neutralizing antibodies. These antibodies bind to the spike protein, which prevents the viral capsid from binding to the ACE2 receptor, and then it cannot enter the host cell. Once this virus is encapsulated or bound to by IgG, it is targeted by other cells in the immune system to be engulfed and then killed. So why are we using new vaccine technology when we've had old vaccines for many years? Well, RNA vaccines, actually the first paper that was successful in the animal model was published in 1990. They're safe. There's no chance of infection from an RNA vaccine. They do not integrate into our cells, so there's no risk of a mutation of some sort. They go directly to the ribosome where they produce that protein. They're very efficacious. They're stable. It causes our cells to produce high amounts of protein that we're interested in, which causes a robust immune response, and they can be modified rapidly. So in the case of a variant that would emerge that wasn't responsive to the current vaccines, the RNA code can be manipulated to simulate that particular mutation that we're interested in. Also, production is very rapid, it's not very expensive, and you can produce a whole lot of vaccine very quickly, which has been very beneficial in this pandemic. This is a cartoon that represents a viral vector, which is this blue sort of star-shaped thing entering a cell. And so, for instance, like with the J&J vaccine, this vector will enter in the cell, it can't replicate, but then the antigens of the spike protein are presented on an antigen presenting cell. In this case, it's a dendritic cell. So it presents to CD4 helper cells, which are both of these cells here, if you can see my cursor moving. What's also important with these vaccines is they did a lot of studies, not just on antibody production, which is done by B cells, which is down here at the bottom. They also looked at CD8 or cytotoxic T cells. These cells are stimulated to find cells that are infected and kill those cells so that they no longer are able to produce virus. And it turns out that all the vaccines that were studied that looked at this had very robust cytotoxic T cell production. So in addition to the antibodies that we make, which are important, we're also stimulating another part of the immune system that is also important in stopping infection and severe disease. So who's eligible to be vaccinated? In the US, for the Pfizer vaccine, anyone above the age of 5. It is fully FDA approved for 16 and over, and that just depends on how much data has been presented to the FDA. The FDA is one of the strictest organizations in the world for approving medications. It does have emergency use authorization for other vaccines and for the 5 to 15-year-old category. And that's done. There are studies that are done, shows that it's safe, but they just don't have the data yet, the numbers to fully approve it. Moderna mRNA vaccine is approved for anyone 18 and older, and so is the J&J viral vector vaccine. There is some differences in the dosing for the vaccines. The Pfizer vaccine is a 30 microgram RNA dose. That's given with each one. The first 2 doses are given 3 weeks apart. And the booster, which also is included for 12 and above. For the 5 to 11-year-old age group, which is now approved for, it's actually a 10 microgram dose. So just a third of what adults are getting in older children. The Moderna vaccine is over 3 times the amount of the Pfizer vaccine. So it's 100 micrograms for the first 2 doses, and the booster dose is actually half of it. It's a 50 microgram dose. The J&J vaccine is a 0.5 milliliter single dose vaccine given initially. Now booster vaccines. So it's now recommended, and this was approved at the end of November, that anyone 18 and older can get a booster vaccine. For the mRNA vaccines, both Pfizer and Moderna, these are given 6 months after the second dose, a minimum of 6 months after the second dose. And for the J&J, it's a minimum of 2 months after the initial dose. And now the booster is recommended. It can be any of the FDA approved vaccines. So if you've got Pfizer, you can get a Moderna booster or a J&J booster and vice versa. I wanted to talk a little bit about adverse effects of the vaccines. Certainly the most common are going to be local, like with any injection, pain, redness, and swelling. Systemic effects can happen too, just from inflammation. These typically last 2 or 3 days, and they do not confer any long-term problems. I have one. Sorry. All right. Early on, when the vaccines were released, there were reported cases of anaphylaxis, severe allergic reactions to the vaccines. We now have over a year's worth of data, and we know that this is actually very rare. Less than 1% of vaccinations given result in anaphylaxis. With the mRNA vaccines, the culprit is thought to be polyethylene glycol. This is used in laxatives like Miralax. With the viral vector vaccine, polysorbate is used to stabilize the vaccine, and it's an emulsifier. It's found in chewing gum, ice cream, whip toppings. It's pretty much ubiquitous in our diet. Both of these would be extremely rare allergies that we see normally in the general population. It was thought that maybe if you had a history of anaphylaxis, whether it be food, venom, medications, et cetera, that you might be at more risk to have anaphylaxis to the vaccines, and this just has not played out. I recommend to all my patients that have had anaphylaxis to any of these things that they have the vaccination, and we've not had any problems personally with any of my patients, and we're really not seeing that on a national level either. Myocarditis has been reported with the mRNA vaccines. This is inflammation of cardiac muscle, which results in chest pain and shortness of breath. This is usually very mild. It does not confer any long-term effects. It's thought that between 1 to 2 of every 100,000 vaccinations given, this occurs, and it's mostly in young males. But we also see myocarditis with COVID-19 infection, and it's a much higher rate of over 10 of every 100,000 infections, and occurs in about 60% of those who have severe COVID infection. It's likely secondary to an inflammatory response to both the virus and the vaccine. Adverse effects with the viral vector vaccines include blood clots. The vaccine can induce low platelets or thrombocytopenia and clots, which is also called thrombosis. This tends to happen in the venous cavities of the brain. It's called cerebral venous thrombosis. This causes a severe headache. It can result in death if there's herniation of the brain. It typically occurs about 5 to 50 days after vaccines. There's one study that was performed that showed it happened in about 2.5 per 100,000 vaccines given. The vaccines have not been linked to any increase in the risk of arterial clots, including heart attacks, strokes. Blood clots have also been reported with natural infection. It occurs about twice the rate of the vaccines, about 5 per 100,000. And we have seen arterial clots happen with natural infection as well. I want to talk a little bit about the age group that's been most recently approved, because I get questions about, should I vaccinate my younger child for COVID-19? Well, these children are just as likely as adults to be infected with the COVID-19, and they do have a risk of severe disease, although it's more rare. In the US, almost 2 million cases have been reported in this age population, and there have been more than 8,300 hospitalizations. These kids are also at increased risk for multi-inflammatory syndrome in children. This is an inflammatory event that crosses all organ systems and can lead to death. And there's been more than 2,300 cases reported. There have been 94 COVID-19 deaths in this age group as of October of this year. And there was a study in pediatrics that looked at specifically at deaths in children and adolescents. And they found that obesity, asthma, and developmental disorders were associated with increased risk of death. So the data from what was submitted to the FDA for the Pfizer vaccine for this age group, more than 3,000 children were vaccinated and monitored for safety. Most common adverse events were local, like with the general population and adults. Systemic effects did occur, but they weren't significantly increased compared to placebo. There was no myocarditis reported, and over 99% of those who were vaccinated achieved seropositive response 1 month after the second dose. So this vaccine is effective in this age population. and does not seem to result in more adverse events. As chromosome 18 and primary immune deficiency relate to each other, I get asked a lot of questions of, if you're immune deficient, should you be vaccinated for COVID-19? And the answer is yes, just about universally. The most common antibody deficiencies that we see, or the most common and primary immune deficiencies that we see with chromosome 18 abnormalities, there are the antibody deficiencies, and by far the most common is IgA deficiency. And this is the most common antibody deficiency in the general population as well. Most patients with this do not have recurrent infections and don't know that they have it. But there is a subset that will have recurrent infections, and it tends to be respiratory infections, mostly sinus infections, bronchitis, and pneumonia. The treatment for it is prophylactic antibiotics. These patients, though, produce normal amounts of IgM and IgG, and they typically respond to vaccines very well and would be expected to respond to vaccination with COVID-19 vaccines. Another antibody deficiency that we see is common variable immune deficiency. This is defined by a low IgG level and a low IgA or IgM, or both can be low as well. These patients have recurrent ear and respiratory infections. They're also at increased risk for autoimmunity. The treatment for it is IgG replacement. Some of these patients can respond to protein vaccines. Universally, they do not respond to polysaccharide vaccines. The most important of which is the vaccine or the infection with pneumococcus or streptococcus pneumoniae. And this is the primary cause of their respiratory infections. If they can respond to protein vaccines, and when I measure or when I'm evaluating a patient with these antibody deficiencies, I'm typically evaluating immunity to pneumococcus, but also to tetanus and other protein vaccines. Most of these patients actually do respond to tetanus. Some don't, but most do. If they can respond to a protein vaccine like tetanus, they should be able to respond to any of the COVID-19 vaccines, because these are all protein antigens. There are other antibody deficiencies that are associated with chromosome 18. Selective IgG deficiency and IgG subclass deficiencies occur. They also have recurrent ear and respiratory infections. Most of these patients, as well as the next one, which is specific antibody deficiency, and that's been described in chromosome 18p minus and ring 18. Both of these antibody deficiencies have an inability to respond to those polysaccharide antigens. And once again, that's pneumococcus. Most of these patients can respond, once again, to protein vaccines like tetanus and should be able to respond to COVID-19 vaccines. The treatment for both of these can be IgG replacement or prophylactic antibiotics. I prefer IgG replacement. And most of my antibody deficient patients that have been vaccinated for COVID-19, when I measure immune responses, they do have positive antibody responses to these vaccines. I had one patient that did not have an antibody response after her second vaccine. She had a booster vaccine that was given. And after that, when I measured, that was positive. Now, if they are being treated with IgG replacement, they are getting passive antibodies from plasma donors. It certainly is possible that if your plasma donor is, and it's going to be multiple plasma donors, if they have antibodies to COVID-19, they are going to give that in the infusions that you get, whether it's IV or sub-Q. And there's no way to measure if the antibodies that you're producing is from your natural immune system making it, or from the IgG infusion that you've been given. So what about the future of SARS-CoV-2? I get asked a lot, is the virus ever going to go away? And of course, we don't know the answer to that. I think it's going to be here for some time, though it's so contagious. Hopefully, as the population becomes more vaccinated, and even with natural infection, we'll see less severe disease and less hospitalizations from positive cases. In the near future, we're about to have oral antivirals that are available. A nucleotide analog, the data has been submitted to the FDA for this, and soon we think a protease inhibitor looks promising. Both of these have shown to reduce hospitalization and severe disease after you have infection. This is typically a tablet that's taken for about 5 days after the diagnosis has been made. And the sooner you take it after a positive test, the more effective they seem to be. There are new vaccines in development that are undergoing trials, and so we'll have more vaccines available. And soon, likely in early 2022, probably the data will have been submitted by the time the trials that are ongoing now, and we will have vaccines available for 6 months and older. That concludes the slide presentation, so I'm happy to open it up to questions. And I have some questions that have already been submitted, and so I'll address those first. And then if any of you have any questions, I'm happy to hear and try to answer to the best of my ability. So one question I had was about COVID vaccination for age 7 years old. And is there anything wrong with tetrasomy chromosome 18 with the COVID vaccines? There's no reported problems with anything with chromosome 18 and COVID vaccines that I could find. So no, I do recommend vaccination to anyone that's eligible for it. There shouldn't be any problem with a chromosome 18 abnormality and vaccination, and specifically with tetrasomy chromosome 18. Another question is, what's been my experience with immunocompromised patients and their antibodies related to the Pfizer children's vaccine? Is the vaccine creating long-lasting antibodies in these patients? So my experience, when I measured early on and people were able to get vaccinated, most of my antibody deficient patients were positive. They were reactive, they had antibodies to COVID-19 after vaccination. As far as long-lasting antibodies, well, the vaccines really have only been available to the general population for about a year now. We really don't know yet. My gut feeling is that some people will have long-lasting and some people will not. I've had some patients that have had natural infection and then recovered from that, and about 10 months later were infected again. And that was through natural infection. And so my feeling is that they lost their antibody response after several months. But I think that's very patient independent, and that's going to be hard to predict on who's going to have long-lasting antibodies and who won't. Another question is, have they been able to test T cells related to the vaccine? If it's not creating long-lasting antibodies, are the T cells still helping to protect? So yeah, the studies looked at T cell response to the vaccine, which looked very impressive, actually. This is a very expensive and tedious thing to do. It is not commercially available to most people. So if you lose antibody response, do you have a long-lasting CD8 cytotoxic T cell to protect you? Possibly. Ideally, you would have long-lasting B cell immunity. After you are vaccinated or you have an infection, memory B cells are created, and these are long-lasting B cells that constantly secrete IgG antibodies and are looking for that same infection over again. My son has very low IgG and didn't respond well to the pneumococcal vaccine, but still has strong immunity from measles that he received many years ago. He doesn't get sick often, even though his IgG is low. He just received the Pfizer vaccine. So I guess that's not a question, but I would expect this child who has low IgG and didn't respond to pneumococcal vaccine, kind of like I discussed, probably would respond to protein vaccines. The measles vaccine is a live weakened virus vaccine, and he responded to that one. And likely he would respond to a protein vaccine, including the COVID-19 vaccines. And that's all the questions that I have that were submitted early on. If there's any more, I'm happy to try and answer.
I have a question, if that's okay. And it's trying to clarify what you're talking about, that pneumococcal vaccine. Is there a reason they don't? Because my daughter is 18P minus and she doesn't have immunity to it. So they wanted to redo it.
Mm-hmm.
Is that something that would help, or is she just not going to develop immunity to that?
So how old is your daughter, if you don't mind me asking?
She's 30.
30, okay. So the reason these patients that are antibody deficient have this problem is that your B cells have to make an IgG antibody to polysaccharides independent of T cells. Okay. They don't get T cell help. Now with protein antigens, they do. T cells are very efficient at directing B cells and cytotoxic T cells when it's a protein antigen, but not so much when it's a polysaccharide antigen. The typical pediatric vaccines that are given in the United States is called Prevnar, or it's PCV13 is the abbreviation, and that's the polysaccharide conjugate vaccine for 13 polysaccharides that we give. There are many patients that I see that don't respond to specifically that vaccine, but will respond to another vaccine that's called Pneumovax, is the brand name.
It's called Niolet?
Yeah, it's abbreviated PPV23, which is the pure polysaccharide 23 vaccine. Okay. There are some patients that don't respond to the pediatric Prevnar vaccine, but do respond to Pneumovax. Okay. I'm assuming if this was done recently at your daughter's age, she probably did not respond to PPV23. That's typically what an adult would be given.
Well, she hasn't had it yet. They wanted to redo it.
Okay.
And I remember from the last time you did this thing that you said something about the pneumococcal vaccine, and that's why I was just trying to clarify. I didn't want her to go do another vaccine if she wasn't ever going to develop immunity to it, because she also doesn't have immunity to hepatitis.
Okay.
So obviously some of her earlier vaccines didn't stick.
Right. So when I see a patient with this problem, typically I do challenge them with that vaccine, because if they can respond to that vaccine, it will stop the cycle of infection and antibiotics that they're in.
She actually doesn't get any colds or anything.
Okay.
She had ear infections when she was very, very young. She had 3 or 4 sets of tubes, but then she outgrew it.
So why did they measure her antibody levels then?
Because we went to an immunologist after she had the COVID vaccine. She had the Moderna. Okay. And after the second Moderna, she had a lot of bruising. She had her period for 3 months straight. It didn't stop. And we also went to a hematologist, and by the time we got into him, they didn't find anything. And so they wanted to test her immunity to other stuff, and they also tested to the COVID to see if she actually got immunity to it after the second vaccine.
So they did, it sounds like a full, probably immunoglobulin or antibody workup on her.
Yeah, and her IgA and her IgG are both low. The IgG is not too low. The IgA is much lower.
Okay. So with IgA, if you're making any amount of IgA, that's considered okay, even if it's below the lower limit of normal. If it's undetectable, that qualifies for IgA deficiency. Okay. You can have low levels of these antibodies, including IgG, and not have any recurrent infections. The way the normal levels of labs are determined is by taking a population without disease, measuring, in this case, IgG antibodies, and figuring out where everybody falls. And what you get is a bell curve. And at some point, they cut off the lower end and the higher end, say the lower 10% and the higher 10%, and this is going to be our normal range. And so we know that even people who fall outside the normal range may not have disease. That's certainly possible. And that may be what you're dealing with. My philosophy personally is to treat the patient, not the number. And so you know that there's a number that's low, But if it's not resulting in disease, which would be recurrent respiratory infections typically, and a need for constant antibiotics, I wouldn't do anything different.
Well, that's why I was wondering whether it's worth her doing the second one, like, because it doesn't seem like she's getting sick all the time. Like, I have more respiratory issues than she does.
Right.
Being older.
This is a conversation I have with my patients, and it's a dialogue. If we want to be extremely proactive, we can go ahead and do it. But if we just want to watch, wait, and see, that's fine. If there are problems that start to happen, then you're going to know what it is. It's recurrent respiratory infections, usually sinus infections, but typically also bronchitis, pneumonia that require antibiotics. If that starts to happen, then you know where the problem is, and that may be fixable with a vaccine. But you don't necessarily have to go and do it if she's disease-free.
Yeah, she hasn't really, and that's why I was like, do we really need to do, because vaccines seem to cause her like inflammatory responses because she already has Crohn's. And so that's why like, if I don't have to do another vaccine, I would prefer not to. I mean, and we're trying to figure out booster wise. What is your thought on mixing Pfizer and Moderna? I know they say it's okay, but I'm concerned.
Yeah. Yes, it's okay. And if patients want to do that, I recommend it. Personally, I've seen more local reactions and inflammatory reactions from the Moderna vaccine. And it's thought to be from the higher dose of RNA that you get. Okay. Now, the booster is not as high as the initial. It's half the, it's 50 micrograms compared to 100 micrograms. But it is more than the Pfizer, which is going to be a 30 microgram for the booster. And so if you're concerned about more inflammatory reactions, I would stick with Pfizer.
Even though she had Moderna, it's okay to mix them?
Yeah, it's absolutely okay.
Well, I'm just concerned about the bleeding, and I never found a reason.
Okay.
But she's in a wheelchair, and just her arms resting on the wheelchair, she had like huge contusions on her arms. So bleeding for 3 months. I mean, it was just obviously not fun.
Right. And that's pretty extensive. That's the most extensive that I've heard. Menstrual abnormalities have been reported with both the vaccines as well as infection. So we know that does happen. I've not quite heard of 3 months of bleeding. That's quite a long time. Did they check her platelets?
She became very anemic. So then we had to do iron and,
So if they did, if they were looking for anemia, then they probably looked at her platelets, and I assume her platelets were okay then.
Her platelets were fine. Yeah. It's her ferritin that gets real low, and then the iron gets all that gets low. And I mean, the hematologist tested over everything, like everything that he could think of, and he kind of researched chromosome 18, and he found nothing in her blood that would have made her bruise like that. So
yeah,
I'm just afraid to do the Moderna again, and so is she. Like, she's like, Mom, I don't want it.
Yeah. So if you do decide to get the vaccine booster, the Pfizer would be a reasonable, or even the J&J vaccine would be reasonable to do. So
most places aren't mixing and matching though. Like if you go to Walgreens or CVS, they will not give you a different vaccine.
Well, it's approved by the CDC. And I can tell you my wife, I'm sure she don't mind me sharing that, she had Pfizer and recently got her booster, and it was Moderna because that's all that was available here. And that was at a local pharmacy.
Oh, okay.
Mm-hmm. So technically they can do it. They may not want to do it, but they can.
Maybe I just need to go in and talk to them, because when you try to book it online, it won't let you do that.
Yeah, maybe so.
Or not, like we're in Illinois and Wisconsin, and neither one of those will allow you to switch it.
Okay. Well, they certainly can, and it's recommended that they can.
Yeah, because I saw people who got Pfizer and got a Moderna booster, they actually had much higher antibody levels, the studies from the UK.
Right. Initially with the Delta variant, it looked like Moderna may be more effective. I'm not sure how that played out over time, more effective than Pfizer. but it was thought to be due to the higher dose of RNA that you're given.
Why is Moderna so much higher? Do you know why it's higher than Pfizer?
I don't know why they selected that particular dose. Normally in the phase 1 trial, in the phase 2 trial, that's usually where they try different doses. And I don't know if they tried a dose that was higher than 100 and just didn't find it more effective, or if they even tried a lower dose to begin with. But I don't know why they went after that particular concentration.
Is there a reason Moderna's not got full FDA approval versus Pfizer that has? Or?
It's going to be the amount of data submitted. I don't know that. I suspect that Pfizer was very aggressive in their trials and recruiting patients, more so than Moderna, and they've continued to do so, which is why they've gotten approval for the pediatric population. Once you get approval for the 18 and up population, you can start doing your pediatric trials, and they seem to be the first on board to do it. And I think it's just probably recruiting and how they recruited for their studies, I suspect. I suspect the Moderna and J&J will fully be FDA approved at some point.
Okay, so you don't think it's anything to do with the vaccine. It's more about data?
I'm sorry, what was that?
You think it's more about data, not necessarily a problem with the vaccine?
Yeah. It's just you have to submit a certain amount of data. The FDA is a very strict and stringent.
Because I think Pfizer came out and said that the Omicron is covered with the vaccine or the booster.
It was a small study. Yeah, I saw that this morning. It was less than 20 patients, I think.
No, that's not very many patients.
To be able to say that early on is difficult. And you know, what was reported early on with the Delta was that it wasn't going to be effective, but actually all the vaccines were effective for Delta. You're actually, when you make an antibody to the spike protein, you don't make an antibody to the whole protein. You make antibodies to individual parts of the protein, and those are called epitopes. There's more than 30 epitopes that we make antibodies to on this protein. So when they talk about a variant, they're talking about multiple mutations in these epitopes. A spike protein is still there, but what we're making antibodies to varies, and that can become different. But if you've made an antibody, let's say, so I think Omicron varies somewhere around like 20 different mutations in the spike protein. So let's just say, all right, it's 30 epitopes that we made. Well, that's still 10 epitopes that you still have valid antibodies for. And that may be good enough to block penetration of the virus into the host cells. It may not, but that's ultimately what decides if a variant is going to be resistant to the current vaccines that we have.
Okay, because I mean, I've known a lot of people that have had breakthrough infections kind of at that 6-month point without a booster.
Mm-hmm.
So obviously it's waning enough and this is Delta that they had.
Uh-huh. So I've seen, and I kind of talked about patients that have had natural infection with, I guess, vanilla, original SARS-CoV-2, and then about 10 months later, probably had Delta. And so their immunity from the first infection waned, I believe. And I believe that because I've also seen patients who were vaccinated who were highly exposed to what I was assuming was the Delta variant because it was in the middle of that wave and didn't get infected. And I made them go get tested, but had no symptoms and had negative tests. And so the vaccines did work for some people against Delta. It's very individualized. You just can't predict how long this immune response is going to last in each person. It'd be great if you did. I kind of feel like in the majority of people, the antibody responses that are happening are probably longer lasting, but in some they're not. And that's the stories that you're going to hear. That's what's going to be reported is that someone was vaccinated and like I said, 6 months later, they were infected with a variant. And that's going to happen. But behind the scenes, there are plenty of people who are being exposed to these variants who are not being infected with it.
Yeah.
And that's the positive side that I'm seeing, you know, and I wish that was reported in the news, but that's not a headline grabber, unfortunately. But it does happen. And
I didn't even think about it that way.
Yeah.
Like there are lots of people, I'm sure, that are vaccinated that are exposed that just.
Yeah, but that's not a news grabber. It's not something you can necessarily prove. You can prove, okay, they were vaccinated and then they got an infection. You can't say, okay, well, they were vaccinated and they were exposed, but maybe they weren't exposed enough and they didn't get infected. So, but it does happen. And I know this because, I mean, these are people in a household. This is a husband and a wife in one instance. You know, there's no way she wasn't exposed to it, but she was vaccinated. He wasn't. And she didn't get it.
Oh, but she tested. See, because the case that I know, it was my younger daughter, her boyfriend. His younger sister was not vaccinated yet because it just got approved. So she got COVID, and they were all together at Thanksgiving. And so obviously they all had to keep testing because she was infected. And it took like 7 days, but the father was positive. Then the mother was positive. But they didn't have any symptoms. So it's kind of like they probably would never have gotten tested except for the fact that their daughter. So I understand what you're saying about the breakthrough then. Like they wouldn't really have known they had a breakthrough infection.
Right, unless they were tested. In the case I'm talking about, we did test. Now, we didn't test every day, but we did test what was recommended at the time, which is after 3 days of exposure.
3 days and then 5 days or something.
Yeah, and had a negative test. And really, that's the success of the vaccine. It may not completely prevent a positive test, but if it prevents disease and it prevents especially severe disease and hospitalization, that is a success. And you really, you know, this is not reported either, but if you have an asymptomatic infection, you're really not shedding virus nearly on the level that someone who's coughing and sneezing that's infected with COVID-19. So you may be positive, but you're probably not spreading the virus nearly as much as if you were unvaccinated too.
But would they still need to quarantine or?
Yeah, we're gonna make them do it. Yeah, that's just standard right now. But the quarantine has gone. You know, at one point it was 14 days or more, and
now it's like 10 or something.
Yeah, now we're down to 10 days, and depending on if you're symptomatic, it can be even less than that.
Yeah, they actually didn't test positive until the 7th day. Like, I think the 3rd day and the 5th day, they were still negative, and then tested on the 7th and they were positive. So it kind of seems like it's a little longer than what they're saying.
Yeah, and potentially it can be. But once again, a patient, someone who has that particular scenario is less a danger going out into society than someone who really is very symptomatic with COVID-19. Because remember, the virus is mainly transmitted in respiratory droplets. It's not truly airborne like measles is. It's typically from saliva droplets, from coughing or sneezing, or even just talking, singing, things like that.
Which I guess if you're not symptomatic, that's not going to spread the same.
Exactly.
Okay.
There should be less virus that you're shedding.
So sorry to interrupt, Nancy. These are all great questions. Just wanted to open it up to see if Kristen or Rachel or Carrie had a question before we close.
I'm good. He answered my question I sent earlier. Thank you. This is Kristen.
Awesome.
Hey,
Thanks,
Kristen. Sure.
Yeah, I've private messaged to my ass, so I just wanted to hear from you.
Awesome. Hi, Andy.
Andy.
And thank you, Nancy, for all your questions. Those were great.
Well, thank you. I appreciate this. It's so informative to, especially get it from the perspective, from the chromosome 18, which obviously we have different concerns.
Sure. Hi, Lewis. Awesome. Well, did anybody have any other questions?
I don't think so. I think he answered all of mine. That was great. I really appreciate it.
Sure.
All right. Yeah, thank you, Dr. Niolet. We just wanted to say thank you so much for taking this time. We really, really appreciate it. And I'll go ahead and pass the video on to a few more members who weren't able to come today, but we really appreciate your time and all this information.
All right, you're most welcome. I'm happy to do it. I hope everybody's doing well and have a great holiday season.
Thank you so much.
Thank you. Bye, bye, Louis. Bye, Andy.
Happy holidays.
Happy holidays. You'll have a great night.
All right, you too.
Bye. Thank you.
Thanks, Holly.
Thanks. Thank you so much. I learned so much, and I know our members are really going to appreciate this. So thank you for your time.
Okay, you're most welcome. All right,
have a good night.
All right, you too.
Bye-bye.
Bye now.